Hello students, a very good afternoon. So today we will be continuing on neural control and coordination. That is the last phase of this chapter where we are going to talk about sensory organs. Okay. So before we start the topic, let me launch a technical poll. So please tell me if my audio and video is fine. Okay. You can answer through the poll, and the YouTube students can put your concerns through your chat box. If there is any problem, then we can look forward to it. All right. So I'll end the poll. If there is any concerns with technical issue, please let me know. All right. So let's go on with the chapter. So today we are going to talk about a very special topic under neural control and coordination. That is our sensory organs. Okay. So when we talk about our sensory organs, so we already know what we are trying to guess. It's about senses, right? So what are senses? Senses are something that gives us an ability to understand our environment in the most logical way. So your senses is an integrated system that includes so many different aspects. For example, imagine that you are that you are in a surrounding. So you try to recognize the surrounding by the noise, by the visuals, right? And by the smell of the streets, etc. So you'll get a profound memory of it. So that is what the speciality of senses is. It gives us an ability to make us, to differentiate the places or differentiate the things that are around us. How would you differentiate a pen from a pencil, right? So that is because you have a visual ability to differentiate the structures and the shapes between them, right? So it is quite complex to even, put it in statements that how integrated our brain and our neural system is to give us this proper sense of feeling. Okay. So we already know that humans are equipped with five senses. So these senses are smell, sight, touch, hearing, and taste. Okay. So today we are going to start with eye and ear in an elaborate fashion, and then we are going to talk about the rest of the senses. All right. So let's go and talk about this. So when we talk about sensory organs, so these are the organs that have the capacity to detect the change in the environment, okay, and convey the information to our, central nervous system, that is our brain and spinal cord, so that we respond to the stimuli that happens around us, okay. So one of the important sensory organs that we are gifted with is the eye, right? So our eye has the capacity for the visual perception. That means that means we have the capacity to differentiate things based on color, shape, okay, appearance, etc. So that is your visual ability. So how many eyes do we have? We have two eyes, right? So if you look at the position of your eyes in your skull, it is present in a socket like structures. Like if you just touch around your eyes, you can feel the bony region around your eyes, right? So that is your skull. So these sockets that are made for the accommodation of the eyes are called as, okay, orbits. Okay, these are called as orbits of eye. So eyes are present inside the orbit where they are held by several sets of muscles, which gives you an ability to move around your eyes based on your interest, for example. Okay. So when we talk about human eyeball, so human eyeball is it spherical? So is it exactly round? Definitely not. Okay. It is nearly spherical. It looks almost spherical, but it is not spherical. Okay. So if we take a cross section of our eye, okay, so this is our eye, right? So if this is our eye, and I will take a cross section of your eye. If I take a cross section of your eye, our eye reveals that it has three important layers, okay? So which makes our eye very functional, okay? So let us talk about these three important layers in detail right now. So eyeball, as I said, it is made up of three layers, and the names of these three layers are called as sclera, choroid, and retina. Okay. So sclera means, see, it's in an order. So we go from the outermost to the innermost. Sclera is the outermost layer, or you can also call it as the external layer of your eye. And since it is an external layer of your eye, it is made up of dense connective tissue. So it basically protects your eyes. Okay. And it gives its, the rigidity as well as the strength to your eyes. Okay. So if you see your sclera region, okay, uh, your sclera region is distributed evenly across the perimeter of your eyeball. Whereas if you see in the anterior region of the eyeball, it is quite, okay, bulged, isn't it? It is quite perforated. So this external region or the anterior transparent portion of your sclera, this is called as cornea. Okay. So the region which is anterior to your sclera is called as cornea. So this is anterior and this is posterior. Okay. All right, people. So in the anterior region, the same sclera is quite transparent and it is quite perforated. Okay. This is called as the cornea. All right. So the next layer sitting very next to your scleroid is your choroid layer. Okay, choroid layer. The one of the important properties of your choroid layer is that it appears blue in color. Okay, so the appearance of blue color is because is made up of many blood vessels. Okay, so what is one important thing that we can observe in the choroid layer is, see, just look at this. If you just take a look at how the choroid, like the blue color here that is present, okay, how it is present, right? So this is the posterior part, right? So this is the anterior part. Just take a look at your choroid. It's thick and thin. So in the posterior region of your choroid, looks very thinner, correct, right? So this is thin in the posterior region, right? So, and it is thick, okay? It is thick in the anterior region, correct? No? So this thickness and thinness is a criteria for us to understand the position of some of the important, important units of your eye. So for example, yes, students, I'm sorry for the inconvenience. So let's get on with where we stopped. So I was talking about the middle layer. So I told that this choroid layer is thin at the posterior region. That's the backside of your eye. It is thin, and in the front side, that is the anterior part of your scleroid, choroid, is thicker. And this anterior part, which is thicker, it forms a special body called a ciliary body. Okay, so when we talk about ciliary body, the ciliary body is something that forms your visible pigment. Like when you look at somebody's eyes, it looks like bluish, brown, black, or whatever the colors you look at the person is because of this ciliary body. Okay, so the ciliary body, it continues to form a visible pigmented region which is opaque. Okay, as I said, the colors of your eyes. So the colors that you can see in a person's eyes, that region is called as the iris. Okay, so what you can find in the iris? So in the iris, you find a central opening, isn't it? There's a black spot when you look at the person's eye, correct, right? So this black spot or the opening of your iris, that is called as the pupil. Okay. Now, whenever you're in dark or your light, you can see that your pupil dilates and constricts, correct? No? So dilation is when you see your person's eyes and it, the black spot that you can see at the center, it would have expanded exponentially. And constriction is like it becomes smaller and smaller as you are exposed to the brighter light, right? So that is because the pupil can change its size. So this regulation of the size comes from the muscle fibers that are associated with your iris. Okay. The iris has muscle fibers. The muscle fibers controls the pupil diameter, and that is how you're able to control, regulate the intensity of light. Sometimes when the bright light hits your eyes knows how to shut it down. Okay. How to control it. So now, after we talk about this choroid layer, one of the most important parts of our eye is the presence of the lens, correct? No. So the lens is a region where the light coming from the outside enters through your eyeball from cornea, and then it passes through the lens, and then it incidents on your retina. Okay. So this part of your eye, which we call it as the crystalline lens, it is present between your cornea. You can see here, this is the cornea and this is the lens. Okay. So this is the region where your the lens are present. Okay. So now if you see your lens, the lens cannot be free floating. So the lens are provided with attachments. Okay. And these attachments are by the ligaments, and these ligaments are from the ciliary body. So what I'm trying to say is, the ciliary body, see, you can see here, this is your ciliary body. The ciliary body is projecting some of the ligaments which hold your lens in place. All right. So that is what gives the firmness to our lens. Okay. So we talked about the outermost, the middle, and now we have to talk about the inner layer. That is the inner part of your eye. So the inner layer of your eye is called as the retina as a whole. So if you see your retina, the retina is not just one common layer, or it's not just one uniform layer. That's what I say. So when we talk about retina is composed of special cells, okay, which we can categorize them into three different groups. So one set of cells are called as ganglion cells. The second set is bipolar cells and the photoreceptor cells. So these three cells, they integrate together for your visual perception. Okay. If you have to see something, it is because of these three cells that will convert this, light rays into an information. Okay, fine. So in these two things, like the bipolar cells and the ganglion cells, first we'll primarily talk about the photoreceptor cells. Photo means light, reception means feeling. Okay. So if your cells have the capacity to understand the change in the light, that is called as photoreceptors. So these photoreceptor cells, they can be classified into two different types. The first type is called as the rods, and the second type is called as the cones. So remember, so these cells are very, very unique because of the presence of the photosensitive pigments that they possess. Photosensitive pigments are some of the proteins or vitamins, okay, or any chemical compound that have the capacity, okay, so that have the capacity to change its structure, okay, based on the incidence of light. Like if the light is put based on the wavelength of light, these photosensitive pigments, they react in a certain fashion. So when we talk about photopigments, okay, or photosensitive pigments, what are they made up of? They're made up of components like opsin, and other one is called as retinol. Opsin is a protein and retinol is an aldehyde of vitamin A. Okay, you might have heard about color blindness, right? So we'll come across like how the retinol is responsible for, retinol is responsible for your night blindness, not color blindness. Okay, so first we will study. See, as I told you, inner layer is made up of three types of cells: ganglion cells, bipolar cells, and the photoreceptor cells. And the photoreceptor cells, we divided them into rods and cones, right? So let's talk about each of these cells, right? So that is photosensitive cells. The first one is the cone cells. Remember, When you talk about the cone cells have the capacity to give the ability to see in light. That means whenever it is daytime, if you are able to see an object, it is because of the cone cells. Since your cone cells have the ability to give you, a capacity to see in the daylight. Okay, this is called as photopic vision. So how do you see colors? It's very interesting, isn't it? So have you seen a TV and they talk about pixels like 1080 pixels? We talk about 4K pixels, 8K pixels, etc. So now you are very popular about it. So how do you know that? What are pixels? Pixels are the basic, fractions of color. So in humans, we have the capacity to differentiate red, green, and blue lights. Okay, so we have three different lights. So these three different lights, when they're incident on different wavelengths, for example, red can have different wavelengths, green can have different wavelengths, and blue can also have different wavelengths, isn't it? So these wavelengths, when they become incident on the photopigments that we talked about, so they produce the combinations, okay, of colors. So that is how you're able to differentiate the different colors that are surrounding you. So when it comes to the other cells, photosensitive cells, that is called as the rod cells. So the rod cells are also called as the night vision. Okay, not definitely night vision. You're able to see things in the dark, right? So, for example, the moonlight or very low light, if you see, you're able to see things at least a little bit. So that is because these rod cells contain a pigment called as, okay, rhodopsin, and the rhodopsin appears almost purplish red in color. So the rhodopsin, it contains no, where do we get rhodopsin? Rhodopsin is extracted through vitamin A. So that is why people who suffer from night blindness are asked to consume, okay, vitamin A rich food, or they're supplemented with vitamin A pills so that they can produce rhodopsin that can help them to see in the dark also. Okay, so if you see, we talked about rods and cones. If you see the inner layer, you can come across, this is called as anterior, correct? No, this is called as anterior, and this we can call it as posterior, isn't it? So if you see in the posterior pole, okay, this is anterior pole, and this is called as posterior pole, okay? So if you see these poles, anterior and the posterior poles, there is a region, okay, which is just above the posterior pole, where you have the entry point for your optic nerves, correct? No, these optic nerves are the ones which carry your eye signals to your brain, okay? And optic nerves collect this information and they leave the eye, correct, right? And your eyes also require blood circulation. So that is why there are also blood vessels that are entering the eyes from outside to the inside. So this point of entrance is just above the posterior poles, okay? So since they enter through this posterior pole, so at this point you don't have any photoreceptor cells, okay? So these photoreceptor cells, when they are not present, obviously you don't have the capacity to sense the light, right? So when you don't have the capacity to sense the light, then obviously you are blind, isn't it? So this part where there is absence of photoreceptor cells, so this we call it as the blind spot. All right, is it clear, students? So next we go across. So if you just move part, you can see an yellowish pigmented spot. Okay, so this is called as macula, okay, lutea. Macula lutea is also yellowish in appearance or yellow in appearance. It is also called as an yellow spot. Okay, so this yellow spot, you can see that they have a central pit. You can see in the eyes, like when you see the appearance of the eye, you can see a central pit, and here the central pit is called as foreway. Okay, the central pit is called as foreway, and the yellow spot, this yellow spot is called as macula lutea. So what is the significance of this foreway? If you see this eye, correct? No, so this is your eye, if I'm not wrong. Okay, so if you see your eye, correct, so you can see that this is your foreway, and at the edges of the foreway, you can see that the retina is thinned out, like the thick retina will come like this. You know what I'm saying? Okay, from thick to thin, it becomes thinner. So at the tip of these thinner regions is the cells that are high in number. So these cells are cone cells. Okay. So what I'm telling, the fovea, right? At the region of the fovea where the retina is thinned out, the retina is made up of large number of cone cells. So that is what gives you a sharper image. Okay. So if you're having the capability of a visual resolution, that is because of your fovea, which is packed with the cones. Okay. So that gives visual acuity or visual resolution. Okay. So another important features that you can observe in the structure of the eye. See, we talked about cornea, right? We talked about the lens. We talked about the iris, right? We talked about the sclera. We talked about the retina, and choroid, and fovea. Now, what is left here? Here, if you see the structure of that is, if you take the section of the eye and observe it, you can come across two cavities or two empty spaces, or maybe the space is filled. Okay. So this space, which is filled by this fluid, okay, there are two spaces that have the cavity which are filled by this fluid. So we are going to talk about these two spaces. Okay. See here, this is your cornea and this is your lens, correct? No? And this is the space that is between the cornea and the lens. So this space is called as aqueous chamber. Okay, this space is called as aqueous chamber. So this chamber is filled by a watery fluid called as aqueous humor. Okay. So another part of your eye is the space between your lens. So this is your lens, and this is your, this is your lens, and this is your retina. So this space between the lens and the retina is called as vitreous chamber. So the vitreous chamber is filled by another type of a fluid, which is a transparent gel, and we call this gel as vitreous humor. Okay, so your eyes is not just empty, it is filled by a gel, and the other portion of the eye is filled by a fluid. Okay, remember, just below your cornea, you have a chamber that is filled with your watery fluid, and just below your lens is a chamber that is filled by a gel-like substances, which we call as vitreous humor. Okay, fine. How do we see people? How are we able to, sense things that are around us? So this visual perception starts from the incidence of light. So you have a source of light. Let us say we have a candle. Okay, we have a candle, and the candle, produces light, and the candle falls on a box. Okay, so when the light incidents on the box, now here the third person. So this is your eye, and the eye is looking at your box. Okay, so how are you able to see this box under the candle light? So the light from the candle hits the box, and from the box it reflects back to your eye. So this is where the visual perception starts. So the light that is reflected from the object, it enters our eyes through the cornea and the lens. So once it enters through the cornea on the lens, here your pupil, it adjusts the focus of your eye, and it incidents a specific wavelength of light onto the retina. Okay, so that means the light that is coming is focused onto the screen, that is the retina. So once the retina gets incident by the light, now this light has different wavelengths. For example, you might say red light, you might say blue light, or say yellow light. So each of these lights have different wavelengths, isn't it? So when different wavelengths of light enter our eyes, they create a dissociation of a protein called as opsin. So when opsin dissociate, so this separation of opsin is based on the wavelength of light, and it creates a change in the membrane permeability. What membrane permeability? That is the photosensitive cells. Membrane permeability will change. So that is the rods and the cones. Once these membrane permeability change, it creates a potential difference. Okay. So this potential differences will lead to the impulse, okay, or the creation of a signal in the photoreceptor cells. So these signals which are produced by the photoreceptor cells, okay, the cells have produced the signals that are the rods and cones. So these signals, okay, create an active potential, okay. So through the bipolar cells, and through bipolar cells, they reach the ganglion cells. And from the ganglion cells, it is carried to the optic nerves. And from optic nerves, it is carried to the visual cortex of your brain. Okay. So it is so beautiful to understand that our eye is not just looking at the objects, it is also giving you the memory of the object that you have already looked at. Okay, the person you have met like 2 years back, or the friend that you met in your lower classes, how are you able to recognize him? So visual impulses are not just looking at the object, it also gives you an analogy and the memory of a particular experience or a particular event. Okay, so that is why vision is very beautiful. Okay, so this is about vision, people. So the another part of the sensory organ, very important organ, is the hearing sensation, right? So when we talk about the capacity to listen to things, okay, so this capacity to listen to things has led to some amazing things as well as the worst things, correct? No, what are the amazing things? Your experience to listen to the sound waves, or your experience to listen to the beautiful music or instruments or voice, or understand people, what language they talk and what is the sound that is happening in your surrounding. It gives you very different awareness when we talk about the ears, right? So hearing sensation is very nice. It gives you exceptional awareness of the surrounding. It balances your pressure conditions. That means you're able to sense the change in the pressure, like how higher the mountain you go, you feel, kind of low pressure, and the deeper deeper the ocean you go, you feel high pressure. Okay, so all these feelings occur because of the speciality of our eyes, right? So let us talk about the anatomy of our eyes. So anatomy of our eyes shows that our eyes can be our ears can be divided into three regions. I'm sorry about it. Our ears can be divided into three regions. So these three regions are specifically differentiated as external ear, middle ear, and the inner ear. Okay. So let's talk about the external ear. See, forget about the humans. Take an example of a wolf or a dog or a cat, okay, or a buffalo. Take a look at their external ear, the ears that you can see in an animal. Can you see that there are different shapes to different animals? Their ears have different shapes. There, some ears are long, some ears are short, some ears are flattened, etc., etc. So this external ear is very, very important. The structure and articulation of your external ear is very important, because the reason why you have these curves and the structures on your ear is because it has the capacity to, concentrate the sound waves that is coming from the further distance deep into your ear, so that you are able to sense the feeling of sound. Okay, that means what is it doing? It is amplifying the sound. Okay, so your external ear consists of two parts. Okay, one is called as pinna, that is called as the earlobe, that what you can see, and the hole that leads to your ear, or it leads to a kennel. Okay, this kennel is called as auditory meatus, or it is also called as ear kennel. Okay, so as I told, your pinna is structured so well because it has the capacity to, concentrate the sound waves, correct? No, so when we talk about the ear canal is composed of, you can see the hairs that are present in your ears, that is interiorly. So these hairs are present, as well as the canal has a coating of a special type of cells, okay, which are modified to form, okay, ceruminous glands, okay, which are modified as ceruminous glands. That is, the sweat gland cells are modified as ceruminous glands. That is the wax you remove when you put the earbuds. So those are formed because of the ceruminous glands. Okay, so these ear canals contain certain cells which secretes a wax, and these wax producing cells are called as ceruminous glands. Okay, why do we have wax and hair in our ears? So because our ears are very sensitive, it has to prevent the entry of any foreign objects like insects, foreign objects like dust, or any of the agents. Okay, so external ear, when you see the canal, right? So this is your ear pinna. So the ear pinna leads to your inner ear canal, and the interior most of your ear canal leads to a specialized structure called as tympanic membrane. It is also called as tympanum or eardrum. Okay. So when we talk about the eardrum or the tympanic membrane, so you can say that this is your outer, okay, this is your outer, and this is your inner. Okay, this is your inner membranes of the tympanum. Okay, outer and the inner membrane of the tympanum. So if you see the outer membrane, okay, so if you see the outer membrane, so usually the outer membrane is formed by the skin. Okay, see, for outer membrane is made up of, it is made up of a layer of skin, okay, which is a semi-transparent membrane. So this is made up of skin, whereas the inner membrane is made up of mucus. Okay, this is called as mucous membrane. So what is the speciality of the tympanum? So tympanum or the tympanic cavity belongs to the middle ear under the auditory canal, correct? No. Now we are coming here for the tympanum, that is the eardrum. This is the eardrum, correct? So when we talk about the middle ear consists of two important regions. One is the tympanic cavity, that is your eardrums. And the second one is ear ossicles, that is malleus, incus, and stapes. Okay. So these three belongs to your, okay, ear ossicles. So let us talk about one by one. When we talk about the cavity, that is the space between, which is present inside the tympanum, it is not just empty. It is filled by the air. Okay. So this tympanum is filled by the air. So when we talk about the empty, it is filled by the air. Okay. So this inside cavity of the tympanum, it is filled by the air. Okay. So this tympanum, which is filled by the air, there is a connection to this cavity from the middle of your ear, is connected to the pharynx. Okay. So this, I'm sorry, students, just give me a second. So there was a network drop. So we'll continue. Yeah. So we were here. So I was saying that, your tympanum is connected to your pharynx through a tube called as auditory tube, or it is also called as eustachian canal. So this eustachian canal gives you an ability to feel the pressure difference. Okay. So that we talked about. So the other part of your middle ear is called as the ear ossicles. So ear ossicles, you remember when we talked about the skeletal system, locomotion and movement, I talked about the skull bones, and the skull bones, we talked about ear ossicles. So ear ossicles, we already know that it is made up of three bones, right? Malleus, incus, and stapes. Remember the names in order so that you can easily understand which one is attached to what. Okay. So if you see malleus, right, this is your malleus. If you see malleus, so where do you see that the malleus is attached to? Malleus is attached to the tympanum, right? So when you see the stapes is attached to what? It is attached to the inner ear, right? So the entrance of the inner ear is called as the oval window. So your stapes is attached to the oval window, that is the entrance of your inner ear. Whereas when you talk about your malleus is attached to your tympanum, okay, which is the end part of your middle ear. Okay. One second. Yeah. So let us talk about your inner ear. So now we are talking about inner ear. So if you look at the inner ear, so the best way you can visualize your inner ear, so this is your middle ear, it's ending. So the oval window will lead to your inner ear. So if you magnify your inner ear image, so this is, one second, I'll just make it clean. Okay, this is your inner ear, correct? No? So if you look at your inner ear, you can see there is two different colors. Okay, you just look at this picture and visualize. So if you look at the inner ear can be divided into two important significant regions. Okay. So these regions are called as bony labyrinth, and other one is called as membranous labyrinth. Okay. One region is called as bony labyrinth and the other region is called as membranous labyrinth. So each of it we have to talk about. So if you talk about the bony labyrinth, so see people, if you see this picture, the purple color that is present, see all this purple coloration is there, no? So this is all bony labyrinth. Okay. So if you see the bony labyrinth, it has a cavity. Okay. So there is a cavity that is present in the bony labyrinth that is called as perilymph. Remember this fluid? The bony labyrinth is filled by, okay, the cavity of the bony labyrinth is filled by what fluid? It is called as perilymphatic fluid or perilymph fluid. Okay. So the other part is called as the membranous labyrinth, right? So when we talk about membranous labyrinth, it consists of two important regions. Both the regions are very, very important. So concentrate. So when we talk about membranous labyrinth, it consists of two regions. One region is called as cochlea and the other region is called as vestibular aperture. Okay. So the cochlea is also popularly known as organ of hearing, whereas the vestibular aperture is called as organs of equilibrium. Okay, people. So remember, the cochlea is called as organ of hearing and the vestibular aperture is called as organ of equilibrium. Remember, so cochlea and vestibular aperture, both of them belong to the membranous labyrinth. Okay, so let's go for the cochlea. So cochlea is a very, very important structure. So that's why we have to study cochlea in detail. So now what we have done is, So this is your inner ear, correct? No. So this coil-like structure, what we are able to see, that we are taking, and we are conducting a cross section. So when we take a cross section of cochlea, we can see some structures like this that is appearing on your slide. So when you look at the cochlea, what you can see, so you can see that your cochlea can be divided. Okay, one second. Your cochlea can be divided into. You can see that it's already visible to your eyes that this cochlea is divided into chamber-like structure. This is one chamber. This is the other chamber, and this is the third chamber. Correct? No? And there are certain layers that is separating these chambers. So if you remember this, it's very easy to understand the structure of or the section of cochlea. Okay. So this coil structure of the cochlea, when we take the cross section, what we can see is there is a upper region. Okay. So this upper region is called as a canal. Okay. There is a canal. So this upper canal is called as scala vestibuli. Okay. This is called as scala vestibuli. And this middle layer is called as scala media. Okay, you can see here, this middle layer is called as scala media. And the inner layer or the lower layer is called as scala tympani. You can see here, correct? No? You can say scala tympani, scala vestibuli. Okay, no? Scala media. So this region is called as scala media. All right, people. So let us go for scala vestibuli. Where can you see the scala vestibuli? See, if you see the scala vestibuli, and if you see the scala media, can you see that there is a membrane that is separating between them? So this membrane that separates between the scala vestibuli and the scala media, it is called as Reissner's membrane. Okay, the membrane is called as Reissner's membrane. So you can see here. So this is called as Reissner's membrane. The upper membrane is called as Reissner's membrane. So the lower membrane is called as basal membrane or basilar membrane. It's very easy. Bottom is base, so basilar membrane. Top, even if you don't remember the name, the opposite of basilar membrane, the only option is coming from Reissner's membrane. Okay. So what is the basal membrane? The basal membrane, now you already guessed it. You're right. So this is the membrane that is separating the scala media. Okay. And the scala tympani, correct? No. So there is a membrane. So this membrane is called as basilar membrane. And this basilar membrane separates the scala media and scala tympani. Okay, people. So now when we talk about scala vestibuli, so this is our scala vestibuli and this is our scala media. Okay. So if you see what is there inside this cavity or inside this canal, is there any fluid or is there filled with air or something like that? So what you can see is these both layers are filled by a fluid called as perilymphatic fluid. Okay. This both is filled by what? Perilymph. Remember. Okay. The middle portion is filled by a fluid called as endolymph. So I'll give you a technique. Outer is peri, inner is endo. So remember, these two are outer, which are this one and this one is outer. So you write it as perilymph. And the middle one is called as endolymph. Okay, people. So when we see the cochlea, we talked about two membranes, isn't it? So among these two membranes, one of the membranes I mentioned was it basilar membrane. You know, the membrane that separates your middle ear to your, scala tympani, correct? No. So this scala tympani separating layer that is called as the basilar membrane. If you see the basilar membrane, so this is the basilar membrane. See, this is your media, right? Scala media. So you can see this is the basilar membrane. You can see this projecting inside the basilar membrane. Okay, scala media. So it is projecting into the scala media. That means it is going towards the scala media. So this projection is called as organ of Corti. Organ of Corti is very important because it is an organ of reception. So this is the one that initiates the reception of sound. Okay. So this is your basal, basal membrane. Now you can take a look at some of the special cells that are present, on the organ of Corti, and you can also come across a very unique type of membrane that is present in the organ of Corti. So let's see what are the structures that are made up of organ of Corti. So what does organ of Corti consist of? It consists of, see, if you say the basal membrane, so above the basal membrane, it consists of rows of sensory hair cells. Okay. So there are cells which produces hair, okay, which are sensory. Okay. So these hairs are called as stereocilia. Stereo means sound and cilia are the hair-like structures. So this cilia has the capacity to sense the, vibrations. So that is why it is called as stereocilia. Okay. So these cells project upward. Okay. That is means outward. So upward, and if you see, the hairs are in contact with the membrane, correct? No, there is a membrane. So these hairs that are projecting from the stereociliary cells, they are contact with the membrane called as tectoral membrane. Okay. So this tectoral membrane, it attaches these hairs to its surface. Okay. So when you see the other portion of your inner ear, so first we talked about cochlea, and the second part of your inner ear is the vestibular aperture, correct? No? So if you see the vestibular aperture, see this is cochlea. Hold on. So this is the cochlear structure, and the rest of the structure is called as vestibular aperture, correct? No? So if you see the vestibular aperture, what can you see? It is made up of tubes, correct? No? Or which is also called as canals. You can see this is the different types of canals you can come across. 1, 2, and 3, correct? No, 3 different types of canals are present. So if you observe these 3 different types of canals, so what can you see? You can see that 2 of the canals are similar, okay, which looks alike, correct? No, so these 2 canals are called as semicircular canals. Okay, these 2 canals which look alike are called as semicircular canals, and they are vertical canals. Okay, they are 90 degrees, whereas the other canals are horizontal canal. Only one canal is present, and this canal is called as horizontal canal. Now, if you see the canal, at the end of each canal is a funnel-like structure, or it opens into a funnel-like structure. So this funnel-like structure is called as an ampulla. Okay, this structure is called as an ampulla. So if you look at the ampulla region, you can find a lump, or that is the bulging. So this lump-like structure near the ampulla region is called as cristae ampullaris. So this cristae ampullaris is a structure that produces long hair-like structures through its cells. So these long hair-like structures are grouped together to form a structure called as cupula. All right. So the other part of your vestibular aperture is the oleanth organs. Okay. When you look at the oleans, how can we recognize oleanth organs? Oleanth organs can be recognized by the presence of, okay, utricell and sacculae. So there are 2 structures called as utricell and sacculae. So they combine together to form oleanth organs. Okay. So this utricle and scapulae, so they project to form a ridge called as macula. Okay. So this utricle and sacculae, they have a ridge, okay, to form a macula. Okay. The cristae and the macula, they are unique because they have the receptors, okay, which have sensory hairs that are responsible for your equilibrium and posture of your body. Like if you're walking straight, if you're balancing from left to right or front to back, okay, you're always in the center, is because of the sensory cells that are present in the cristae and the macular regions of your vestibular aperture. Okay, fine, people. How are you able to hear? Okay, let's make it quick. So the sound waves coming from the outside, it reaches your ear pinna, that is the external ear, and it travels through the ear canal. Okay, so this ear canal, it ends at the tympanic membrane, and the tympanic membrane starts vibrating because of the pressure that is applied by the sound waves. So these vibrations, they reach the inner ears through oval window, or inner ear ossicles through oval window. So in the oval window, what happens is you have the perilymph region. You remember? So this perilymph starts vibrating. When the perilymph starts vibrating, the hair-like structures which are in the, which reaches the tympani, that process, that basal scalar tympanic membrane starts vibrating, and the structures that basal membrane, they start the tectoral membrane. Once they start pressing against the tectorial membrane, now once this gets excited, correction causes the nerve impulse, and that's you hear this sensation work again. Sound outside sounds ear. Okay, as sound waves. I'm extremely sorry, students. Just give me a second. I'll share the screen. Okay. Yeah. So hold on. Yeah. So, what was I saying? So once your vibrations ovular window, now this vibration starts occurring. So these vibrations makes the tympani to apply pressure on the basal membrane. When the basal membrane moves, so you remember that the hair-like structures are present on the structure of the basal membrane, correct? No? See, what did we talk about this membrane? That is the organ of Corti, right? Organ of Corti has hair-like structures. So these hair-like structures press against the tectorial membrane. Once they press against the tectorial membrane, these sensory cells get excited and they form a nerve impulse, and the nerve impulses are carried to the auditory center of your brain, and that's what causes hearing. So what are the other senses you have? The other senses you have is the touch, olfaction, and gustatory senses. The touch you already know. Olfaction is your sense of smell, and your gustation is a sense of taste. Okay, so let's talk about your olfaction. Olfaction is your capacity to sense the smell, and your capacity to sense the smell comes because of your innermost layer or outermost layer of your nose is coated by mucus coated receptors called as olfactory receptors. Okay, the inner lining of your nasal canal is lined by cells called as olfactory receptors. So these olfactory receptors contains epithelium that has the capacity to bind to the chemical molecules that are in the air as smell. When the specific chemical bind to these olfactory receptors, they create a certain change that will stimulate your olfactory neurons, okay, near the olfactory epithelium, which are outside the environment, okay. From there, it directly travels all the way till your uh, sorry, it all the way travels to your olfactory bulb that is present in a broad bean size structure in the brain. Okay, so in the olfactory lobe, the senses that are perceived because of the excitations of this olfactory epithelium or olfactory receptors that will stimulate your limbic system. So I already told you limbic system is a part of your brain that is responsible for your emotional excitation. Okay, so if you're emotional or if you're expressing emotionally, that is because of your limbic system. All right. So the other part of your sensation is your organs of taste. Your capacity to taste different types of foods and enjoy different delicacies or even get disgusted by different types of food is because of the receptors that are present on your papillae of your tongue. So if you look at the mirror and just open your tongue wide, you can see that there are, pimple-like structures on your tongue, correct? No? So these bulbs that are projecting outside your tongue are called as the taste buds. The taste buds, okay, usually these are called a papillae. So there are different types of papillae or circumvallate, foliate, fungiform, as well as filiform papillae. So these papillae have specific taste buds which are gustoreceptors. Okay, gustatoreceptors or gustoreceptors. So gustoreceptors means capacities to sense the taste. So when you chew your food, the chemical molecules bind to these gustatory receptors, and via supporting cells, they create an impulse. Okay, so that creates a sense of taste. So the sense of taste as well as the sense of smelling is almost similar. Okay, and these similarities are judged by the same limbic system. Okay, which does both the jobs. Okay, the limbic system is responsible for both your olfactory and gustatory reception. And skin, coming to the skin, it is one of the largest sense organs in our body because the amount of area the skin covers. So it contains receptors which are have the capacity to sense heat. Correct? No, you are able to sense the change in temperature when it gets cold, or when somebody touches you, or when something hurts you, or somebody holds you. Right? That is the heat, cold, touch, pain, and pleasure. All these senses are equipped in your one of the largest organs, that is your skin. All right, students. So here we come to the end of your neural control and coordination. So let us solve some of the previous year questions, okay, to understand better. So here is your first question. Okay, Needles bodies are mainly composed of. Your options are proteins and lipids, DNA and RNA, nucleic acids and smooth endoplasmic reticulum, free ribosomes, and rough endoplasmic reticulum. Okay, so I launch a poll for 1 minute. Okay, so you can answer the question faster. Okay, it's very easy. We discussed it already. So I want you all to answer in 30 seconds because it's a very direct question. All right, students, it's already time. I'm going to end the poll now. So let's see what's your right answer. So knee cells bodies are basically composed of free ribosomes and free ribosomes and rough endoplasmic reticulum. So these are the regions which are very uh, engaged, okay, which are highly engaged in protein synthesis, which are responsible for neural stimulation. So your answer is D, that is 4, okay, that is free ribosomes and rough endoplasmic reticulum. So the second question is here, which of the following structures or regions, okay, is incorrectly paired with its function? So here are the options. Medulla oblongata controls respiration and cardiovascular reflexes. Limbic system consists of fiber tracts that interconnects different regions of the brain, control movement. Hypothalamus, production of releasing hormones and regulation of temperature, hunger, and thirst. Corpus callosum, band of fibers connecting left and the right cerebral hemispheres. All right, students. So here's a question. So let me launch a poll. Okay, I will launch a poll for you. So here is your poll. You have 1 minute to answer this question. Read the question carefully and answer this question. Okay. It is a very easy question if you have remembered the functions of the structures we have already spoken about. Okay. So I will end the poll right now because it's time. It's very direct question. We can just go into conclusion directly. See, if you look at the options and if you remember the topics that we have taught, the first option, that is the medulla oblongata, correct. It controls respiration and cardiovascular reflexes. Okay. Whereas when it comes to limbic system contains fiber tracts which are interconnected to different regions of the brain, correct? Okay, so here limbic system, it does not control movement, it controls emotions, right? Your anger, fear, okay, happiness, all of these expressions and emotions are coming from the limbic system. Okay, so the second option is wrong, whereas the third and fourth option, that is hypothalamus production of releasing hormones and regulation of temperature, hunger, and thirst, this is also right. Whereas corpus callosum is a region that connects your left hemisphere to the right hemisphere of the brain. Even that is also correct. So our option is option 2. This is a wrong option, right? Limbic system is emotional center. It's not controlling the movement. Okay. So next question. The transparent lens in the human eye is held together. Okay. Give me a second. I'm not able to see. Okay. Is connected or held together, uh, or it is connected by the place, or it is connected by what? Okay, ligaments attached to ciliary body, ligaments attached to iris. Okay, hold on, people. Let me read the question properly. Uh, okay, fine. Ligaments attached to its iris, smooth muscles attached to the iris, smooth muscles attached to the ciliary body. Okay, so we are talking about transparent lens in the human eye is connected, or it is held in place by what? Okay, so I will launch a poll again, and you have 30 seconds to answer because today we discussed and it's a direct question. Okay, so let's go. All right, students, I'm going to end the poll now. So the transparent lens of a human eye is held together by the ligaments that are extended, or it is formed, or it is attached to ciliary body. So the option one is correct. Okay. So remember this, ciliary body ligaments attaches the lens. Okay. So next one. Receptor sites for neurotransmitters are present on. Your first option is membranes of synaptic vesicles, presynaptic membrane, tips of axons, postsynaptic membrane. Okay. So you have 1 minute to solve this question. I'm going to launch the poll now so you can answer. Students, your polls are live now. You can answer the question in a minute. All right, students, your time is up. So let me end the poll now. So let's see if you have got the right answer. So receptor site for the neuron transmitters are present on. So you already know, right? The presynaptic neurons, they send the signals, and the signals are through the neurotransmitters, and the neurotransmitters go and attach to the receptors and the postsynaptic neuron. So the postsynaptic neuron membrane has the receptors which come and detects these type of neurotransmitters. So your option is option four, that is post synaptic membrane. All right, students. So it's time. So I'll see you in the next class. Have a nice day. So if you have any doubts, you can post your doubts on the chat box or in the next class. I'll see you in the next class. Okay. Thank you. I'm sorry for the technical difficulties.